Is that the microphone on now, hopefully? Hello, everybody. Oops, sorry.
No, it's loud.
So the microphone's on. If you could all just take your seats, please. It's my great pleasure to introduce to you our keynote speaker for this afternoon's session, Angela Bosco-Lauth, who has a post at the University of Colorado. She's an assistant professor there, and she did a lot of the very early work demonstrating the susceptibility of different species to SARS-CoV-2. So we very much look forward to hearing the latest from your lab, Angela.
Thank you so much, Margaret. All right, well, welcome back, everyone. I hope now that we've all had some coffee and warmed up a little bit, hopefully I won't put you back to sleep. So it's been a pleasure to be here. Ireland is lovely, and some of you who may have heard me talk in Scotland last year, some of these slides may be familiar, but I do have some updates, so it won't all be old hat stuff. But I wanted to just talk a little bit about some of the experimental work that we've done at our lab with SARS-CoV-2 since the onset in companion animals, domestic animals, and wildlife, with pretty an abrupt transition to wildlife in the last year or two, as that's become more of a theme, at least across the US. So no surprise to anybody here after several really elegant talks this afternoon that SARS-CoV-2 is clearly a zoonotic pathogen. And it became obvious very early on that cats, at least big cats, if not domestic cats, were probably going to have some sort of an impact, given that we saw cats infected in zoos, and then people's homes. Those reports started to follow quite quickly. And of course, the mink story is familiar to everyone. Why mink are more susceptible to clinical disease than ferrets is a mystery, I think, still to this day, but certainly has something to do with housing and also probably some host receptor specificity. And then the deer story has been really interesting, and you'll hear a lot more about that tomorrow during the wildlife session, but I'll have a few things to say about that today. So my lab mostly does experimental infection studies in animals. We're fortunate enough to have a large animal BSL-3 facility, and we do a lot of work with other coronaviruses, namely MERS-CoV, as well as early work with SARS-CoV-1 as well. So we were pretty well set up right from the very get-go in 2020 to start answering some of the basic questions about species susceptibility. And what we typically do is we do kind of a very I guess, simplistic model where we expose animals to the virus by whatever the most natural route of infection that we can simulate would be. And in the case of SARS-CoV-2, that was going to be an intranasal inoculation. And then we do clinical observations and sample collections for a period of several days or weeks, and followed by analysis, interpretation, and then that leads on to further studies to kind of analyze what else is happening. So some of these further studies include transmission, a little bit of environmental sampling as well, and then also thinking about evolution in these species. So our approach to the different species really has to do with what's available, for one thing, what's most interesting from a perspective of what the world is looking at. So again, really early on, we realized that dogs and cats in people's homes were becoming infected. There was some evidence of clinical disease in those species. And then we have some historical reference as well, because SARS-1 was actually cats could be used as a model for that disease as well. We also then looked at livestock. If you can think back to 2020, it was a big question in everybody's mind is what happens if we have, an infection with a disease like this that spreads rampantly through a herd. That could have had devastating consequences. And I think, fortunately, 3 years later, we can say, phew, that didn't happen, thank goodness. But we did some of those experiments early on to just kind of tease out what the inner workings of this virus might be in different species. And then our approach to wildlife is a little bit surreptitious. Some things are, well, what can we get our hands on? And then there's also reports of things that have been found in nature. So again, the white-tailed deer, for example, there was experimental work and serological work that supported why that might be an interesting species to look at. And then you start thinking about, well, what's related to white-tailed deer? What other cervids could we think about as well? And then peridomestic mammals, I'll use that term quite a lot. So that's just referring to any wildlife that might live in and around human inhabitants. So that's usually things like field mice, deer mice, small rabbits, sometimes skunks, raccoons, animals that you frequently would consider maybe pests, or at least those that are sharing the same feed bin as perhaps your pet cats might be. And so clear back in March 2020 was our kind of first call to action to try to figure out what's happening in domestic animals. And so we started off with very small sample sizes. We used pilot studies where we just had 3 animals that we would inoculate and just kind of see what happened. And so we started off with just dogs and domestic cats. We inoculated them intranasally and then followed them doing a series of sampling, collecting rectal swabs, oral secretions, nasal flushes, as well as blood, and just to look and see, do we have viral shedding? And if so, do we have any clinical disease? And also, what's our immunological response? We also collected tissues for histopathology, so we could check and see if there were things that were happening subclinically in cats and dogs as well. What we found really early on is that there was very, very little, if any, evidence of clinical disease in these animals. We certainly didn't see that. Other studies did report that with the early strains, but our studies did not see clinical disease. But what we did see was some pathological disease in cats, nothing at all in dogs. We also did see quite a lot of shedding. So to answer a question from earlier, cats are pretty good little viral replication machines. And so this is, again, using these kind of parental strains. Clear back in March 2020, we only had one variant at that point in time. So this is using the Washington 01 strain. And we saw really high titers of cat viral shedding in the nasal and the oral cavities. So that immediately made us think we probably had some transmission capabilities at least, so we should consider looking at that more. And we typically start all of our experiments by looking for infectious virus. One, because we can, that's what having a BSL-3 is really nice for that. And also because at that time, I don't even know that primers had quite been designed and identified as being really exceptionally good for detecting this particular virus. So we stuck with the infectious virus titers and then later compared them to PCR. And so these are just a couple of images from the pathology in these cats. And as a caveat, these are somewhat older cats. These cats were between 5 and 8 years old, so I'd consider them fully adults. And we did see some interesting lesions in cats that were clinically very normal. We didn't have any weight loss, no fever, no respiratory signs, but there was evidence of some rhinitis and some inflammation in those lower airways as well. So here we have some alveolitis in cats that, again, had no clinical disease. And this was taken from animals that were euthanized actually at 2 time points. So early on in the course of disease, after 5 days, we saw more evidence of some pathological change in the upper respiratory tract. And then later on, the 28-day phase is when we saw animals that had some of the more alveolar changes. And if you look at other models that we've since used, which is primarily going to be the golden hamster, you tend to see a lot of acute changes early on, and then the lung changes can actually happen quite a bit later, even once they've resolved shedding and resolved any clinical signs. So it's not really surprising, but at the time, without any evidence of disease in these animals, this was a little bit interesting to us. So we followed that first little pilot study up with just a cat-to-cat transmission study to see if cats could in fact transmit to each other. And these animals were group housed, so this doesn't answer the question about aerosol, it's more about direct contact. And what we found is that when you commingled inoculated cats, you really readily got secondary infection in the contact animals. And with these cats, it was within less than 2 days of commingling, we started to see transmission and really high shedding profiles, just like the ones that we directly inoculated. The good news, at least early on, is that cats that were re-exposed, after 28 days of infection, developed no clinical response, no virological shedding, and had a really nice boost in antibody titer. So these early strains have a very high antigenicity and a very high neutralizing antibody titer, which is probably somewhat contrasting with Omicron, as we heard earlier. So early Washington strains and even Delta, these cats develop a very nice response that are probably protected for some level of time, if not months, maybe even a year or so. That's probably changing now as we're seeing strain variability and things that are less immunogenic in cats. Oops, that's weird. Somehow the title of that came off. But we also did a study where we compared adult cats versus juvenile cats. And this was relatively interesting, where we had kittens that were less than 13 weeks old. We used the same, we used 3 different levels of viral inoculation in kittens and subadults compared to these adults. And we found that the younger the cat, the less clinically relevant and or virologically relevant they were for shedding. And so in these little guys, we had really low levels of shedding. Only about 1/3 of the animals actually had infectious virus shedding, and it was higher with higher titers of virus that was inoculated. So when we did like a 10 to the 3rd, that'd be our low level of inoculation. Those cats didn't shed at all, but they did seroconvert. And then cats that were exposed to our peak titer, which was 10 to the 6th, they did in fact shed, but the duration was 2 to 3 days versus adults where it's closer to 5 to 6, and the titers were generally lower. In the subadults, we saw something right smack in between kittens and adults. So still lots of viral shedding, about 1 log to 2 logs lower than what we saw in the fully adult cats, and the duration was 1 to 2 days shorter. So with cats at least, geriatric cats were a little bit more likely to be sustaining an infection and potentially transmitting. We also didn't see any pathological evidence of disease in any of our kittens or subadult cats. So they seemed to clear infection readily, develop a beautiful antibody response, and we didn't see any evidence in either their upper respiratory tract tissues or their lungs of pathological disease. And I believe, thinking back again to the earlier strains, that was very similar in people, where there was kind of this, like, kids were relatively not susceptible to those early strains, middle-aged adults probably were the worst, and then in other cases, older adults were also quite affected as well. So I believe this has also changed with the changing of the virus, and I'm not sure if we would see the same results now if we did this with Delta or with Omicron, but at least with Washington strain, it seemed to be that kind of older adult cats were a bit more likely to be the ones that carried on and maybe even transmitted forward. So that all kind of predated up to what we tried was a vaccine trial for a company. This was published by Merck. This was basically before we really thought that cats were going to be, well, I guess before we knew what was going to happen with cats, there was a lot of interest to find vaccines that could be used for animals. And in fact, I believe Zoetis had a vaccine that was being used in zoos as kind of a donation, essentially, to help protect big cats. That may even still be ongoing, I'm not sure. But we did a vaccine trial for this company early on to see if vaccination in these cats would prevent infection, and in fact it worked beautifully. So what we found is this was actually not quite sterilizing immunity. We still had some PCR positive cats following vaccination, but no viral shedding. And vaccinated and infected cats did not transmit virus to any contact animals. So really quite an elegant vaccine and quite lovely, and you can read more about that. But this was a vaccine that was replicon based, so not an mRNA vaccine, but one that's been used in other animals as well. So great, wonderful, exciting news for us. And then obviously the last couple of years have shown that most people aren't all that concerned about vaccinating their pets, so this really hasn't hit the market, but it was a good proof of concept. So kind of the take home with dogs and cats both is that clinical disease really isn't that common. There definitely are exceptions to that rule, and I'm sure any vets in the audience have probably seen a handful of cats that have come into your clinics that have respiratory signs. They may or may not be SARS-CoV-2, but usually I think if you have a patient that's got SARS too, and their cats get some respiratory a little bit later, you probably can make some assumptions about that. We tended to see that cats were much more susceptible than dogs, just in general. I didn't show you the dog data because it was frankly really boring. Our dogs didn't shed any infectious virus. They had very low PCR positive results for a couple of days and had very, very low seroconversion, such that I don't think they would be protected from reinfection. But again, dogs don't seem to be a really problematic species for this transmission chain. As was already mentioned several times, cats are almost certainly getting this from their human owners. There has been one case, again, of a cat that has transmitted to a veterinarian. And to my knowledge, that's the only case I'm aware of pet cat to human transmission. So it is still a possibility, and I think most veterinarians are well aware of this at this point. So, if you know you have an infected owner and you're not already wearing a mask to protect yourself from the owner, perhaps think about their cat as a potential source of transmission as well. But in general, I think the story with cats and dogs is that humans are still much more likely to give each other COVID than our pets are. So still back in 2020, with the original parental strains, we also decided to undertake some testing with livestock species as well. We used calves instead of adult cattle, because it's just frankly a little bit easier to work with in a BSL-3. And we tested goats, sheep, alpacas, meat rabbits, and one single horse, which I know is not really livestock, but they don't get classified as in any one category. And what we found with these animals was also really not exciting, thankfully. We didn't see any infectious virus shedding in oral or nasal secretions or rectally from any of these animals. We did have one calf that had positive tracheal tissue. This was early in the course of infection, so that could have just been some local replication. There was nothing in the lungs, it was just in the trachea. And so we take that as kind of a interesting, but probably so what, you know what I mean? And we also found that these animals didn't seroconvert readily. So in general, either low to no antibody titers in any of these species, which indicates that they're probably not really seeing a productive infection. Even if we get a handful of PCR positive samples early on, it's possible that virus is just kind of petering out, no cells there to replicate it robustly, and no immune response to speak of. And in 2020, this was good news, and now that we've seen 3 years of a pandemic and no real evidence that there's any problems in livestock, I think we can almost put this to rest, at least for the time being, barring, and I'll say this with fingers crossed, any crazy mutations that may happen. So again, take home with livestock, we're not super concerned about it. Maybe I'm saying that prematurely, but at this point I think with livestock in particular, we have other much more serious pathogens to worry about, things like avian influenza and African swine fever. So there's always something that we can be concerned about, but I think we can put SARS in the back burner for now. So wildlife has been kind of where we've taken our turn, and again, this is a caveat to which you'll hear more about tomorrow. But we started off with doing some projects with that, again, parental strain to just look at what these peridomestic species might be doing in the landscape. What if you have mice in your home? Could they be contributing to the source of infection, et cetera? And so we undertook to take quite a few of these little species, things that were easy to find or that were readily available or that we know are going to be around human homes, and we started testing them with that same basic experimental design. So intranasal inoculation followed up by sampling, whoops, and then pathogenesis and transmission if necessary. And I have some kind of oddball species on here. Armadillos I stuck on here because they're one of those weird creatures that have a very low body temperature. There was some evidence from another paper that there was a PCR positive from some location. And of course, armadillos have leprosy, which is just weird in itself. So we decided to test armadillos just to see what happens. Cottontails are kind of a species of least concern that are all over the place, at least in the US, and I know all over the place in other locations as well. And then we looked at a lot of these mesocarnivores, and I particularly wanted to look at skunks because they're closely related to other Mustelidae species. And so the idea that if a mink and a ferret could be exposed and you expose a skunk, probably going to have some similar predisposition. We looked at coyotes and red fox as being good candidate surrogates. And then possums, again, strange little species that are frequently found in and around people's homes. We also looked at Mexican free-tailed bats. I think early on especially, there's still lots of evidence that this virus came from bats, but then the fear, of course, is that what happens if it gets back into the bat population, especially in New World bats? Are we going to have to worry about a whole new virus emerging? So we wanted to undertake that. And then lastly, I'll talk just very briefly about cervids, and I'll make the caveat that this is unpublished data in the cervids, so I'm going to talk about it and then answer a few questions about it, but I don't have explicit permission to give all the details on that yet. So here's kind of our laundry list of different species and the strains that we use to inoculate. Most of these, we started off with the Washington 01 strain, as a lot of this work was done before there were other variants out there. And then we went back and repeated a few of these species that were interesting with Delta and/or Omicron. And so what we found right off the bat is that most of these rodents are actually not super susceptible, with the exception of deer mice and wood rats. And deer mice have been published on a few times now. Why deer mice are so different from mus musculus is a little bit of a mystery to me, but we found that they were readily infected. They shed nice levels of virus for a pretty short amount of time, and clinically they're relatively unaffected. So our particular findings didn't see any histopathological changes. We didn't notice any sort of weight loss or respiratory distress in our mice. They all survived, but they did shed quite a bit and had a nice antibody response. And the same could be said for the wood rats. So of the rodent species we looked at, those were the two that were interesting. Squirrels were thankfully negative, as were prairie dogs, and our house mice also were negative. You'll notice on house mice, we tested for both Washington 01 and Omicron. If you recall, I think last year, right when Omicron first came out, there was some evidence that maybe it was a virus that had from human to mouse and back into human. Maybe that was where that mutation took place. We did not find that to be the case. At least the mice that we tested were not susceptible at all to Omicron. So that doesn't necessarily disprove that theory, but for us at least it put to rest that we had to worry about house mice at this point. But what's probably most interesting and not really unexpected was that skunks are readily susceptible to both Washington 01, Delta variant a little bit less so, and not at all to Omicron. So, and when I say susceptible, what I'm talking about really is viral shedding. And in this case, we also did not have any PCR positive skunks when infected with Omicron. These animals were PCR positive even when they weren't shedding virus for the other two strains. So skunks, I would say, are kind of lowly susceptible. So some animals will shed virus, but not all of them do. And what we did find is that skunks did not readily transmit to other cage mates, or excuse me, roommates. They were actually group housed. So skunks are interesting. Much like with the mink and ferret situation, we now have another Mustelidae species that has a very different response versus the mink. So I think there's lots of differences in probably receptor specificity, location perhaps of the ACE2. There may be other differences as well, but typically I don't think we're going to see a whole lot of skunks causing problems on the landscape with SARS-2. What I thought was really interesting here is the red fox. And so when we did the Washington 01 strain, all 6 foxes shed. They shed actually relatively high titers, close to what we'd see with cats. And then the same with Delta. So red fox, being a canid species, I was actually somewhat surprised by this. I expected them to behave a bit more like dogs. And you'll note that coyotes were completely negative. We did have a couple of PCR positive samples, but it was very, very high CT scores, so nothing that we were super concerned about. So why foxes are different from other canid species is a bit of a mystery to me, but this is a replicatable experiment. So we've now done 3 different rounds with foxes, and every time we can get them infected with Washington and Delta. And then foxes will directly transmit to close contact cage mates. So that's interesting as well. I think in terms of what we're seeing in the landscape, this is probably a species to maybe keep an eye out for, but unlike deer, foxes don't tend to run around in giant herds, so I think lots of transmission in the environment is probably unlikely. But one thing that I think about is what about predatory chains? And if you've ever had a cat go missing in an area where there's lots of coyotes or foxes, that's maybe a potential for a interspecies transmission event to take place. And then last on our kind of susceptible species list are the mule deer. And these are some of the most recent studies we've done. So we actually went ahead and just skipped the Washington 01 strain since that hasn't been in circulation for over 2 years now. And we used Delta for deer. And this was last summer, so at the time this was actually right before Omicron really took over. And Delta was what we were seeing most frequently in deer that were being isolated in the landscape. And so we did our Delta inoculation and found 5 out of 6 deer to be susceptible. And I will put the caveat here that one of the deer that didn't shed was actually a contact animal. So we had 4 directly inoculated and 2 contact deer. And one of our contact deer did not become infected, but the other one did. So we now have evidence of mule deer that are becoming infected and transmitting to each other. And if you're not familiar with North American species of cervids, it's totally fine. Most people actually aren't even in North America. So North American species that are most common are white-tailed deer, which you'll hear lots about tomorrow. Mule deer tend to be a bit more geographically distinct, so we tend to see them more in the Intermountain West, which is where I'm from. And they will sometimes interbreed with white-tailed deer. So they're not genetically all that distinct. So the reason that I don't think we see a lot of mule deer that are popping up positive all over the place is that there just simply aren't as many of them around. And also they tend to be a little bit less likely to come up into people's yards and eat out of their hands or out of their cattle feeders. So mule deer are just a little bit more shy in general. But again, quite susceptible. Interestingly enough, the elk, which are, again, a cervid species, but really not that closely related. We didn't see any evidence of viral shedding in those guys. We had 1 or 2 that had PCR positive samples in the first day or so after inoculation, but that could just be residual virus, and they didn't seroconvert. Or excuse me, they did seroconvert, but not to the same level as the deer. So elk are probably not going to be something we have to worry about in the future as well. And then lastly, we tried our possum and armadillos as kind of our little oddball species, and thankfully none of those were infectious or susceptible to infection. So these are the species of wildlife that we've seen that do have some level of shedding and could potentially play a role in the landscape, particularly if we start to think about if the virus starts to mutate and change again, maybe these would be the first ones we continue to surveil and make sure we don't have any weird things popping up in our wildlife. And just to give you an idea of kind of what that level of shedding looks like, I know these graphs are kind of hard to see, but this is in log PFU. So these would be our little rodents up here. So that's actually a pretty low level of shedding in both rodents and skunks. This is barely above the limit of detection of our assay. Compare that to the cats, which were in the 5 to 6 log range, which is about more like what you might expect from a person who is infected. This is a pretty low level shedding, may or may not be something that even would be transmissible to, say, a person in the same room. So we think probably more direct contact transmission, especially with these low titers. And then these are the foxes. So foxes shed really nicely from both oral and nasal routes for up to 5 days, maybe even a little bit past. And the foxes have a tendency to shed to that kind of same level as cats. So again, this is a species that I think we probably shouldn't discard altogether as not being important. I don't think they're going to see a whole lot of red fox in the landscape that are a problem, but foxes can certainly get into and around human properties quite a bit and may be a source where you can see some of that spillover or spillback taking place. And then lastly, the mule deer shedding is shown on this graph here. Again, mule deer shed really quite high titers as well. So this really isn't surprising. This would look very similar if I had showed you whitetail. But they did tend to shed for a little bit longer. And so this might be why we're seeing deer on the landscape as a species that can propagate transmission within their herds. So something about deer just makes them a little bit more likely to shed virus for longer periods of time and to maintain it in their own populations. So kind of my take-home message for wildlife is that there are quite a few species, at least, that we know are susceptible based off of experimental work. Other labs have done lots of work. I think you're going to hear about badgers tomorrow. So it's going to be an ongoing thing that I think largely the COVID world is interested in. Again, mink, primates, big cats tend to be a bit more severe, whereas our domestic mammals, a little bit less severity of disease. And a lot of these species don't have any disease, but may propagate infection, be a problem for any sort of potential for evolution down the line. And so that's kind of the last thing that I just will mention. This was a study that we did in our cat populations, where we just looked at what the evolutionary factor was within the virus that was being shed from those cats and compared it to the inoculum strain. And I will mention briefly, I think somebody talked about this morning, that early on we figured out that there was a lot of mutability when this virus was passaged in just cell culture. And so we did compare this 3 different cell culture passages, and you do see quite a lot of strain variability even just within those passages. So we used an early a single passage viro isolate of SARS-CoV-2 to do this particular study, and still found that with cats in particular, you reach fixation of mutation relatively quickly. And so this D614G was one of the first mutations, even in the human population, that we started to see it was fixing itself relatively quickly. And so one cat transmitted this particular mutation and it was fixed in another cat. So that just kind of demonstrates that those species that are the most susceptible to infection are also probably the most likely to have a mutation that then sticks within that species. If you look here in dogs, ferrets, and hamsters are a really good model of infection as well. But in particular, like the dogs, you see this really high level of variants that are produced. And again, these dogs didn't actually shed infectious virus. So this is just what's being seen in kind of the RNA that's been pulled off of PCR. So what's probably happening here is that you have some cells that are replicating very, very minimally, and that virus is really not taking hold. So you're probably seeing a lot more mutation frequency and either ending up in sort of missense, nonsense, or just virus that peters off altogether. So what's next? So the next steps right now, I think, are going to be to just basically continue on with that the evolution study. So our current program is looking at all the different viruses that have been shed from our wildlife species, especially those that we're able to transmit, so the foxes, the deer, I believe that's it for now, and look and see if we see mutations within those species and see if those mutations come to fixation relatively quickly, or if it's something that you see a lot of variability. We have very small sample sizes, so those numbers are not going to be as exciting as they could be, but with any amount of luck, we'll be able to make some predictions about what to look for as we move forward in the landscape, especially with deer. And so again, recommendation for pet owners, veterinarians alike, be careful with cats, and in general, just be more cognizant of going out and working with wildlife. I think bat people in particular were very, very cautious because they didn't want to transmit SARS-2 to bats. And as we all have known, that caution has kind of dropped off a bit with time. But I do think when we handle wildlife, we should be very, very cautious about transmitting to them and vice versa. And with that, I have a laundry list of people to acknowledge, as does everybody, but I'll go ahead and take any questions if we have time. Thank you.
No, you didn't. No, we definitely have time for questions, and I'm sure there'll be a few. Alan, do you want to go first?
Lovely talk. So we work a lot on defining the permissivity of animals based on the receptor usage, so more from a molecular perspective. And I was just interested in that table you had where you were looking at the percentage of animals that had been experimentally infected and then the antibody positivity. Mm-hmm. So I guess my sort of somewhat simplistic way of looking at that might be to assume that any animal that's ACE2 is not efficiently used, probably won't get into very many cells, not generate very much antigen, and therefore not seroconvert. Do you think that's broadly true, or would you challenge that and say maybe there's cases where you're getting just fine, but for some other reason there isn't a long-term antibody response? Sorry, very long question.
That's a great question. I wouldn't challenge that. I actually think you're right on the money, although I'll be honest, my knowledge of the molecular characteristics of the ACE2 receptor are minimal, so I would definitely look to you for more answers about that. But I tend to think that you're probably right. It seems like the species that were less susceptible in general didn't seroconvert and didn't have a high, even PCR replication efficiency. So to me, that's showing that we're not getting good antigen presentation from whichever cells are being infected in the first place. So that's, I would tend to agree that I think it probably has to do with ACE2. There may be other receptors that are also playing a role there, but it seems for antibody specificity in particular, I think if we're missing that major receptor binding event, I don't think we get a good serological response. Yeah.
Thanks, John, and then
You'd shown with your cat studies that kittens, adults, and geriatrics were different in the way they were infected and how much they shed, et cetera. For your studies with the wildlife, what ages were those? Because we've seen drastic differences between juveniles and geriatrics.
Yeah, that's a great question. Most of our wildlife species are adults, and of whatever age we caught them at, which is a great question also. But no, that is a really good point. The only other thing I can make a comment is that when we look at like hamsters, the younger the animal tends to be a little, like very, very young hamsters maybe shed a little bit less than the geriatric animals early on. But I haven't been able to really tease out those kind of variations in the wildlife species because we just don't know how old they are. We try not to use juvenile animals whenever possible because of that exact variability. Good question.
Really nice talk. Thanks a million. A quick question and a longer question, I guess. The red fox, obviously in the UK and in Ireland, urbanization of the red fox is massively increased over the last decades. Is that something that we're ignoring in terms of screening for a reservoir of SARS-CoV-2?
Potentially. And I can only speak to what I know about in the US, and that's that our populations have actually decreased a bit, so I don't think people are quite as, well, maybe they are interested. I should leave that question to the USDA folks. But it would be a species that I think would be interesting to look at if for no other reason than that we know how susceptible they are and because of how frequently they come into contact with our pets.
And with the longer question, I guess, is the transmission or the lack of evidence of transmission from cats to humans, or limited, very limited transmission. How do you explain that? Because you're getting very high levels of virus coming out, infectious virus being shed. Is there some change in the virus, or is it the fact that the primary transmission is human to cat and the humans are already infected so they won't get reinfected? How does that work? What's the dynamics there?
Yeah, I think what you said secondarily is exactly true. I think most cats are getting it from their human owners, and therefore that there's probably no transmission back because I think cats will shed for less time usually than people, and we better have at least a few days of immunity, right? So, I really think that's the crux of it. And why we don't see more cat-to-cat transmission, possibly because we're not looking for it. I've seen a couple studies where within households, cats can transmit to each other, but I think in general those people have already been exposed, so we don't see a whole lot of that going back into the humans. Good questions.
Just a question as well. I really love that talk. You caught these animals. Have you screened them for flaviviruses and parasite loads, because that could affect things as well? Have they been screened?
Oh, yeah. Great. So we typically only screen the wild-caught animals for other coronavirus. Everything gets screened first for coronaviruses, so we just do a general neutralization test on those. And then I should have mentioned that our skunks, our foxes, our raccoons, the deer, and the elk, those are actually all captive herds, so they already have disease screening processes of their own, but I haven't screened them for flaviviruses. It's actually not a bad point to try that. Just look for other things that might be contributing. So good point.
Hi. Thank you very much for the interesting talk. I was just thinking about red foxes and such. Could the likes, since they would kill things and potentially be shedding, could maybe the likes of crows and scavengers like that there be a reservoir to look at, but considering they being potentially ingesting this and being in close contact with this? I know they're birds, but still, maybe it could be an area to look into as a reservoir and another potential method of transmission.
Yeah, I think the scavenger question is really important. I think when it comes to birds, I only put up there a couple of different species. We don't have any evidence that in general birds are going to be susceptible, so I wouldn't worry about crows or even things like vultures, but other scavenger animals certainly I think could be a problem. We do have a study plan that I haven't quite figured out how to undertake, which involves feeding deer mice to other susceptible species. Our IACUC doesn't love that concept, so we have a little bit of work to do on that. But I do think it's an important question, because if that predation or scavenging could be a source of propagation or onward transmission, that could be a place that we just haven't really nailed down yet. So yeah, great point.
Fabulous. Thank you
Great.
so much, Angela.
Thank you.
That's wonderful. And if I could pass over to my co-chair, Wim, to introduce our next speaker. Thank you.
Thank you, Margaret. My name is Wim van der Poel of Wageningen University in the Netherlands, and I'll introduce the next part of presentations for you. And the first speaker is Alan Radford of the University of Liverpool, and Alan is going to speak about canine enteric coronavirus. Go ahead.
Thanks very much, and thanks for the organisers for letting us talk about a variant. We use the word variant now. We never use the word variant for coronaviruses, but I think we just all use it now. We use the word variant for a variant of canine enteric coronavirus, which we've been monitoring, if you like, in the UK, and its association with a very changing pattern of gastroenteric disease in dogs. Hands up if you've got a dog at home. Okay, well, a little bit lower than the usual percentage, but we all know dogs vomit, right? Dogs vomit once and then eat it and are fine. You know, that's what dogs typically do. They don't often get very ill, especially if they're vaccinated. But this is a new pattern of disease which we've been noticing in the UK, where dogs are really very severely ill. 2 or 3 days of consistent vomiting, very lethargic, not eating, and a few more days before they get better. The virus we're interested in is canine enteric coronavirus. It's an alpha coronavirus. It fits in down here. That's kind of blurred for me, so it probably is for you, but it's very closely related. Can't move from the microphone, to FIP and to TGEV, which we've heard about before. Interestingly, there are recent variants of canine enteric coronavirus discovered in humans, very rare, but I guess people aren't looking for them. Typically, canine enteric coronavirus would be associated with very mild diarrhea. There is actually a vaccine for it, but the vaccine companies can't sell it. It's been around a long time, causes mild diarrhea or inapparent infections. That's the normal dogma. There are case studies of very severe disease. They're usually puppies. They're usually co-infected with other viruses. So in the UK, in 2020, remember 2020? So here we go. This is February 2020. Local vets were talking about a different pattern of disease associated with severe vomiting, something that they hadn't experienced before. It was a very complicated time for us, because this was just before, this is SARS coronavirus 2. So it was just here, just before things were really kicking off in the UK. There was some concern, maybe it's COVID. How to respond to this as a country is quite difficult, because there are no national surveillance groups or organisations for companion animals in the UK or in any country, I think, in the world. So these sort of anecdotal reports from vets in practice are usually quite difficult to respond to. What we've been doing is developing a national surveillance system for companion animal disease in the UK, based on 2 things. 1, electronic health records, so a very different kind of science to what we've been hearing about today, and 2, based on enhanced diagnostics and sequencing. So what we were able to do using electronic health records was to look at gastroenteric disease in about 10% of the vets in the UK in real time. We have about 10 million records, and I don't know if you can see up here, gastroenteric disease is normally gently seasonal, but in winter 2020, something very different was happening. Very different was happening. It was very specific to gastroenteric disease. This is respiratory disease, no change. This is cats, no change. This is the use of a drug to treat vomiting in dogs, and you can see it was being used much more by 10% of the vets in the UK. So we were pretty sure something different was going on. When we looked at in diagnostic laboratories, so these are commercial diagnostic laboratories, there was one virus that stood out, which was canine enteric coronavirus. It had a similar seasonality to its diagnosis. That's the blue line. Nothing different about 2020 to previous years, but a similar seasonality. So my colleague Shirley, who's sat over there, decided to test some samples for canine enteric coronavirus. Highly premature, in my view, but she did it, and she was absolutely right. So this is M-gene PCR, and this is whole genome sequencing. We're a very small team, but we managed to get through that process, led by Shirley, in just a couple of months. And the end result of that was that there was a variant of canine enteric coronavirus. All these viruses had the same sequence. This is the whole genome sequence. Their whole genome was identical, and it was spread across the UK. So a very different pattern to what we would usually associate with canine enteric coronavirus. That was 2020. Life moves on, 2022. The similar thing was happening, mystery dog vomiting virus. This was being reported on the east coast of the UK. People were worried that it was some toxin in the water. Again, we got some samples this time from vets in practice. 40% were testing positive for canine enteric coronavirus. We also got some samples from diagnostic laboratories. 30% testing positive for canine enteric coronavirus. When we looked at the sequence, so what we were interested to see was this the same variant as from 2020? The bottom line is no. So this is the 2022 variant here, shown by these sequences. Again, the dark purple, you can see it's spread Scotland, Northern Ireland, all the way down to the south coast. This was a widely dispersed variant of the virus, but it was different to the virus that we saw in 2020. That's based on the M-gene PCR, and we looked at the whole genome. What we found was that, so the query here is the major variant from 2022. It was very similar to the major variant from 2020, apart from in the spike gene, where there was clear evidence of significant recombination. The ancestor of that we never found. Interestingly, the most closely related sequence to it was a published sequence that's been on GenBank for a long time from the States. So we don't know the origin of that recombinant, but a recombinant nonetheless. What we've come up with the idea, if you really want to understand the evolution of this virus, you have to look at two levels. You have to look at the spike gene to understand recombination, but you also need to look at the whole genome to understand the relatedness. If we just looked at the spike gene, we wouldn't understand that this virus did evolve from the variant in 2020. It did, but by recombination as well. So I mentioned briefly the identification of canine coronavirus variants in humans. Again, rare. This group and this population from Haiti, I think it was mentioned this morning in the context of a large animal coronavirus. But they found canine enteric coronavirus in these people too. But these human canine enteric coronavirus variants have deletions, which I think are in the N-terminal domain. So the viruses that we have in dogs, we suggest are not a zoonotic risk at the moment. What are we doing in the future in terms of monitoring this? Well, we've put together a sample of Canine enteric coronavirus is collected over a full year, so we can see how this virus is progressing from summer into winter. There is a lot of variation. This is M gene PCR, and the yellow viruses are the ones from this current season. And we're just picking which ones of those to whole genome sequence. We're using health records to monitor the disease in the dog population, this severe gastroenteric syndrome, and these data are publicly available on a website. And you can see very clearly here that pre-COVID, there wasn't really much of a seasonality. We then had a big spike before COVID, that's our 2020 outbreak. And then we're left with fairly regular winter seasonality, which we think is associated with canine enteric coronavirus. We're trying to isolate the virus with a collaborator. We would love to show Cox postulates that these variants are associated with more severe disease. We haven't done that, so we use the word associated with, not caused by. And then just very briefly, some very weird different science for this audience. We're also using machine learning of text to see whether we could spot outbreaks like this in the future. This is led by a colleague of mine called PJ Noble. And the long and short of it is topic modeling and artificial insemination. AI, I'm getting my AIs muddled up. Artificial intelligence would indeed spot this outbreak fairly reliably. So if you want to know more about that, just ask. So to conclude, there is a new pattern of gastroenteric disease in the UK. We're pretty sure of that. It's severe in some affected dogs. It's associated with, not caused by yet, but associated with canine enteric coronaviruses that are evolving. Don't need to read that out. So this system of national surveillance for pet animals is quite neat. Happy to talk about that with people afterwards. And then fake news. So I got asked about this by the local newspaper in Liverpool. We have to be very careful how we talk about coronaviruses at the moment, don't we? And I was very, very careful. I spoke to this newspaper and I said, yes, it's a coronavirus, but it's not COVID, it's not SARS. It might be related, it's a distant cousin, I think was the phrase I used. It doesn't affect people, and this was the headline. So be very careful what you say. So lots of people involved on the data science and statistics and epidemiology. This is PJ, who does the machine learning. And then Shirley, who does all the wonderful sequencing work for us. This work was funded by Dogs Trust. Thank you.
Okay, thank you, Alan, for an interesting presentation. Are there any questions? Okay, in the back.
Thanks for the talk. I just wonder, have you looked outside the UK for cases of it? Like in Southern Ireland, for example? I know I just did a quick Google and there's lots of chatter from previous years about something very similar to what you were describing. So I guess it has travelled.
Lots of chatter, but I mean, hands up if you've got a national surveillance system in your country for companion animals. It's the difficulty is moving from chatter and anecdote to... we did try to get some samples from the Republic of Ireland, but haven't managed. So if you've got a way of, didn't try very hard, but if you've got a way of doing that, that'd be great. And if you are aware in your own country of similar patterns, because there should be, right? We're not expecting this to stay confined to the UK.
The clinical signs, Alan, are they only vomiting or also diarrhea?
Yeah, vomiting, diarrhea, very lethargic, very unwell, 2-3 days. But I think a real spectrum. I mean, right, we hear the anecdotes for the really sick, the animals that are sickest. I think lots of animals probably have a fairly mild or even inapparent infection. And a lot of animals that get gastroenteric disease in the winter have something other than canine enteric coronavirus. It's not the only virus that's involved.
Thanks. I just had a question. I guess it's like dog epidemiology, but so where do you think the variants are coming from? And how quickly does a variant, do you think, spread in dog populations? Presumably they're not that well connected. I mean, maybe at a local neighborhood level, yes, but then a kind of national level, how quickly could a variant spread, do you think?
Yeah, I guess we don't, with the level of sampling we've done, I mean, we're not sampling at the level of human populations in SARS coronavirus 2, right? We have hundreds of samples, not tens and tens of thousands of samples. I think dogs are pretty well connected. I think, you know, perhaps people have got some experiences. There's a lot of mixing of dogs locally at parks. People will travel more widely at the weekends, more widely, not wildly, widely at the weekend. So maybe that is one route. We think the winter thing might be to do with the survival of the virus outside the animal. Dogs do have a unusual behavior, if you know, when they greet each other. So there's quite a good way of transmitting a fecal pathogen that way, through sniffing feces, sniffing the bottom of their friendly dog. But yes, it is curious that it has this national distribution so quickly. But it's whole genome sequencing, so I think it's reasonably reliable, that result.
Question over there.
Maybe not, but just in relation to the last question, I mean, it's like 30,000 a month of imports from Eastern Europe, and often pretty grim health conditions. Could that be related to the input and spread and peak?
Yeah, potentially. We probably wouldn't know about severe gastroenteric disease in those countries. I think it's a bigger conversation, isn't it? This movement of dogs globally that is happening into America from Asia and into the UK and Europe from Eastern Europe and further afield. We're moving viruses around and other pathogens in dogs in a way we never used to. And it's this particular habit of rehoming, very well intentioned, but quite dangerous, bringing animals into a new environment, a new country.
Okay. Okay, if there are no more questions, thank you, Alan. We now move to a few so-called flash talks. Actually, we have two. There are three in the program, but Mr. Morozan is not here, so we have two flash talks. So these are short presentations, and there is some room for questions after the presentations, especially because we have two instead of three. And the first is by Marsha Wallace of the University of Oxford and Royal Veterinary College about innate immunity in canine respiratory coronavirus. Please, Marsha, the floor is yours.
Hi, you may remember me from earlier. I was talking about FIP in cats. So this is a flash talk that essentially is the parallel to that, but what we're doing for dogs, canine respiratory coronavirus. Very similar to what I was telling you earlier about with the cats. Canine respiratory coronavirus, highly contagious. This is beta coronavirus. It's particularly problematic in kennels. And similarly, we have a situation where most of the dogs, they have mild clinical signs. They recover without treatment. They're just fine. It's a very, rare, small percentage of the dogs that end up developing, fatal lung disease and major complications. And so we have this question as, who are these dogs? Why is it fatal in some of these dogs? What's really going on there in terms of, for example, host genetic susceptibility? And so much like we designed this overview for the cats and FIP, we have the same kind of a setup for the dogs, where we're looking at a variety of different breeds of dogs. We walk through the same kind of process where we're looking at whole genome sequencing, and then really, because we're working with these small cohorts, high noise environment, and can only follow up a select finite number of target variants in a larger cohort, how do we rank and prioritize those variants? And so this is a system, a bioinformatic system that we've been using, again, applying from our complex diseases that we were looking at, and now applying that to coronaviruses. And so we're able to then take those candidate variants and follow them up in larger cohorts. And come see the poster. The crux of it is kind of here in that we're identifying some really interesting variants that are in genes that are involved in pathways tied to innate immunity and the Wnt signaling pathway. Thanks very much.
That was quick. Well, there is room for some questions, and there is a poster also, as I understand. So, are there any questions? No? Yes, Alan.
I guess the important thing is the dogs that you choose as having severe disease. Do you test them for other pathogens to make sure they've just got respiratory coronavirus?
Yes.
It could be quite complex.
Yes, and that's actually something that we're tracking quite carefully, and especially moving forward with our larger cohort. One of the things we've got is the status of several different diseases that they can be co-infected with, and so that's going to be, we're going to be expanding that type of analysis, yes.
Okay, is there, okay, there's one more.
Just a quick follow-on to that. Do you have the, I guess, longer-term clinical history? I wonder if maybe some earlier infection predisposed them to getting exposed to the respiratory virus and it becoming severe, or how far back do those records go?
Yeah, that's a good question. Lucy probably knows more, but I'm actually not sure what clinical information we have on those dogs prior. I know that a lot of the information is collected from the kennels, so we have from that point onward. I'm not sure what exists for prior. Lucy's going to answer that.
Hi, yeah. This is a kind of serendipitous study in a way. It's done in a very large rehoming center where dogs arrive, and typically if you arrive in the rehoming shelter, you seroconvert to canine respiratory coronavirus within 3 weeks. And dogs at that shelter get rehomed, but they also get euthanized, and they get euthanized for a variety of reasons. Sometimes they get euthanized with really severe kennel cough. So actually what we've done is come along 20 years later, actually, in the middle, during the pandemic, and thought we should resuscitate those samples now with the tools we have and see, can we tell who did well and who did badly just by looking at their genomes. So we have very intense history for them for however long they've been in the shelter, but nothing before that.
Okay, thank you. Okay, then we're done with this presentation. We move on to the second flash talk, and that's by Beatriz Tomeo-Martin of University Alfonso X in El Sabio in Spain. And Beatriz is going to talk about SARS-CoV-2 in pets. Please take the mic.
Thank you very much for your kind presentation. Good afternoon. As mentioned in previous talk, we know that several animals that are susceptible. Wait. Here.
It would be about 30 seconds before the, no, you didn't do anything. The projector's on a timer. So 4-hour timer, sorry. It will be about 30 seconds before it lets me turn it back on.
Okay, that's all.
It's all right.
Oh.
Luck on the draw. Just have to wait till this stops. Okay.
I'm going to take the opportunity whilst we've got a little pause. So tomorrow we've got the workshops in the afternoon. So we would like the people who are helping us coordinate the workshops to stay back after John's invited presentation. So hopefully we'll just have a very short meeting of the people who are coordinating. And that includes Cyril and Valeria, who are organising the workshops tomorrow. For the vaccine and therapeutic group, it's Louise, Dallin and Zoltan. For diagnostics and surveillance, it's myself, Margaret, and Dan Horton, if he arrives. And for epidemiology, it's Vim, Richard, and Rachel. If we could just have a quick 10 or 15 minutes to sort ourselves out for tomorrow, that would be. And oh, Alan's taking. Alan didn't know that, but you've now been nominated. Thanks, and thank you.
No. Okay. Again. As mentioned in previous talk, we know that several animals are susceptible to SARS-CoV-2 infection, and that has been related to contact with infected human, particularly in pets. That's why it's important to know the potential risk habits associated with owner-pet transmission and whether pets contribute to COVID-19 pandemic. Regarding this, today I would like to share you our main findings about this new coronavirus in companion animals in Madrid, in a region of Spain. We have carried out an epidemiological study in 87 pets from a household with COVID-19 people. We performed a questionnaire for identify the risk habits, and we collect swabs and blood samples to evaluate SARS-CoV-2 and immunoglobulins against it, and also in some dogs, antibodies against other virus. 18 cats and 69 dogs were included, and the variables studied were sex, weight, contact with people, contact with animals, type of feeding, owner jobs, symptoms. No. The descriptive analysis evidenced that 7 males and 9 female cats from different age groups and different type condition were included. More than half of them were fully vaccinated and slept with their owners. Many cats were in contact with other people, and only 30% in contact with other animals. On the other hand, 20 males and 41 females dogs were analyzed, and also they were of different age groups and different type condition, body condition. Sorry. In this case, about 85% were fully vaccinated, and only around 20% slept with their owners. In this case, 80% were in contact with people and also with animals. In both groups, around 45% of the owners were healthy care workers, and the percentage of pets with antibodies were 28 for cats and 30 for dogs. No cats saw respiratory symptoms. However, 14 dogs saw we did. About approximately 25% of both groups presented digestive symptoms. Attending to the association of variables, we found association between dog symptoms with weight, vaccinated status, contact with people, contact with animals, and type of feeding. And also, we found association between cat respiratory symptoms and healthy workers' owners. And regarding humoral response, we found an association between anti-SARS-CoV-2 antibodies and anti-infectium canine hepatitis antibodies, canine parvovirus antibodies, and canine distemper virus antibodies. Finally, the key insight of this study, where the SARS-CoV-2 seroprevalence is slightly higher in dogs than in cats, close contact with positive owners appears not to be determinant for higher immune response, and although we have found some association, we haven't found risk factors that favor the development of anti-SARS-CoV-2 antibodies. Thank you.
Okay, thank you. Time for some questions.
When the pets showed clinical signs, did you have any evidence or information about how long it was between when the person was sick and when the animals got sick?
I don't know if I understand your question. Do you say that if we measure the antibodies previously and...
No, so you started by going to households that had people with COVID-19.
Yes.
So what was the time between their diagnosis and the animal clinical signs?
Okay. We choose the household with at least one people with COVID-19 by PCR, evaluated by PCR. And then we take their blood sample of the pets and evaluate again by PCR and antibodies by ELISA. I don't know if I know.
Maybe how quickly, so when a person tests positive, how quickly did you know, were able to go to the household and test?
Yes. How soon after the testing of the human person did you test the cat?
It's a good point. I try to the time between the both samples were low. However, we can't assure that the time is less than 1 month. Thank you.
Okay, thank you. Any other questions? No? Thank you.
Thank you.
And thank you for these 2 presentations. Then we move on to our final presentation, and that's Dr. John Hardam of Cerritos. And this presentation is sponsored by the Microbiology Society. And the presentation is about COVID-19 zoonosis, mitigating the risk of susceptible animals. John Hartman is director of the Zoonosis Center of Transboundary and Emerging Diseases, and he led multiple projects on the development of vaccines for companion animals. John, please take the floor.
Thank you. Well, it's a pleasure to be here this evening, this afternoon. And I also recognize that I'm the one standing between everyone and break and dinner. So we've heard a lot about the companion animals in various species. I show a number of articles up here that demonstrate the public's interest, especially when zoo animals started to become infected, and there was a number of very highly publicized deaths in zoo animals, most especially in snow leopards here. But as we've heard, there are a lot of different domestic and wildlife species that are susceptible to varying degrees to SARS-CoV-2. One of the things that we're very interested in is the One Health approach to veterinary care and its interrelationship to human health and environmental health. And that is definitely the case with SARS-CoV-2. It is a very good example of the importance of One Health. We've heard a number of cases of human to animal, but just wanted to highlight a few of the human to animal to human cases that have been publicized. We heard a few of those earlier, and I think Angela brought up a couple of those. The human to mink to human transmission in the Netherlands was very highly publicized and resulted in a number of countries actually culling all of the mink in their areas. some cases up to about 15 million mink were culled because of this. Human to zoo animal to human transmission. This is a publication by Audrey Segrest at Potawatomi Zoo in Indiana in the United States. And this was a case where an infected lion infected several of the zoo staff. This is a case of the human to hamster to human transmission that occurred in Hong Kong for the reintroduction of Delta variant back into that region and a number of cases that resulted. Of course, we heard about the questions around the origin of Omicron earlier, so I won't cover that anymore. And then was also brought up about the human to cat to human transmission in Thailand. So a number of cases there, and then the very interesting cases of the highly divergent variant that arose in the deer population in northern US and southern Canada. Whereas Omicron had about 32 mutations, this deer variant virus had 76 mutations away from the Wuhan 1 strain. And it was shown to be transmitted, in some cases it's thought, from human to mink to human to deer to human. So a number of possible jumps there. The underscore, though, is the importance of One Health, protecting the animal population to help improve human health as well. This was the case I mentioned at the Potawatomi Zoo. The publication by Audrey Segrest. It was a geriatric lion infected from unknown source. They think it may have been from a carrier, one of the zoo staff. But while the lion was being assessed on the table, it coughed in the face of some of the zoo staff, and they got multiple transmissions off that. You can see here, the contact with the lion is the circles here. So 3 different staff had contact with the lion. The lion started showing symptoms, very acute onset of illness here. 2 of the staff had contact at that point. Very shortly after, they developed symptoms. The 3rd staff had contact with the lion out here, and then very shortly after, they had symptoms. A lot of sequencing was done. You can see the phylogenetic tree here. So you can see the sequence of all of these individuals here is the same as compared to sequences in the surrounding community. So it's a very nice paper showing the reverse zoonotic and then zoonotic transmission in this case. The paper in Thailand showing the feline to human transmission. This was a case where humans were infected. The cat that they were traveling with was separated from them and ended up infecting some of the healthcare staff shortly thereafter. And you can see in this case also the sequencing showed all these isolates from the patients, and the cat was all identical as well. As far as symptoms in the veterinary world, very similar to that we see in human cases where symptoms are actually present. A number of the symptoms that have been reported in human and veterinary world are shown over here. Obviously, the number of veterinary patients that show symptoms is much, much smaller. This is a wonderful website you can go to, SARS Annie, to see the variety of different species that have been infected. It doesn't really show up, but this is actually the world map in different regions. Again, age dependency of symptoms. Variants behave different than Wuhan 1, as we heard earlier. And many of the early susceptibility studies were done with juvenile animals in Wuhan 1. Obviously, subsequent to that, people started looking at older animals, and as we heard from Angela, many of the wildlife were in adult animals. And then we have the impacts of the variants. Going out from the original Wuhan 1, Alpha, Delta, Gamma, on around to all the different Omicrons. So it's important when you're looking at a vaccine to assess the ability to cross-protect against this wide variety different variants. Now, when the first reports came out of dogs and cats being infected, we began a vaccine program. We have a number of different coronavirus vaccines that are on the market of different varying types. For this one, we opted to go with a stabilized spike recombinant protein. It is formulated with a proprietary aqueous-based adjuvant system. And our recombinant protein is a stabilized spike that is soluble. You can see the schematic of the stabilized spike trimer here in this cone shape, and an electron micrograph of our recombinant protein. You can see this nice cone shape with an extended tail, and the crystal structure of the protein matches the shape of our recombinant protein as well. Our initial studies we did assessing a serologic response in dogs and cats as part of an adjuvant selection study. And you can see here the red is the control group, the orange was a different class of adjuvant system, and the green is our aqueous-based adjuvant system. And you can see after one dose, we got a moderate and variable response. After 2 doses, we saw very, very high levels of serum neutralizing antibodies. As the story with mink was evolving, we pivoted and looked at mink as well, in both doing laboratory serology studies and also a field safety study in a mink farm. And we were able to demonstrate very high levels of serum neutralizing antibodies generated as a result of vaccinating mink. We then turned to look at this vaccine was based on the Wuhan 1 spike protein. How did it interact with the variants? In this case, you have the Delta variant shown here, and then the Omicron variant with all 32 mutations shown. We used a pseudovirus assay to assess the ability of serum raised following vaccination to neutralize the different variants. In this case, you see the Wuhan 1 titer and the Delta variant titer at day 0, 20, and 42. And you can see we have comparable, statistically non-significant though, difference between neutralization of the Wuhan 1 and the Delta variant. Now, we were able to use the vaccine and provide it to a number of mink farms in the US, Canada, and some in Europe. But that was when the story was evolving in zoological animals. We'd had the opportunity to provide a small number of doses to the San Diego Zoo after their primates became infected, working with the USDA to get what's referred to as a 103.3 permit, or an emergency use permit for an experimental product. The zoo vet community is a very small community, as I found, and they talk to each other. So after providing the vaccine to the San Diego Zoo, we started getting barraged with requests for vaccine from other zoos, especially after the reports of deaths in snow leopards started to occur during the Delta wave. So we began a vaccine donation program. Our initial programs were with the experimental vaccine that had not yet been licensed. And that was on a case-by-case basis that was permitted by the USDA. So a lot of administrivia associated with it. We started out donating about 11,000 doses of experimental vaccine, and then in round 2, about 16,000 doses. And this was 224 organizations requested and received the experimental vaccine, and you can see the locations where those requests came from all around the world. We were able to get feedback from many of the vets as part of that permit process, and also from the mink farms, where we had about 3.5 million doses that were utilized. So over on the far side here, we have some of the feedback from the minks. We saw no significant adverse events. No major safety observations reported, and some very good feedback that we received. No mink farm that vaccinated their animals had an outbreak. So that was very nice news on efficacy. In terms of the zoologic vaccination, about 11,000 doses and 16,000 doses. We did have what we refer to as significant human adverse events. That's just a exposure. So a lab technician was trying to vaccinate an animal. The animal moves and they stuck themselves or scratched themselves with the needle. In all three cases, there was no issues with the handlers. We had 0 significant adverse events reported for the animals and less than 1.5% minor safety observations. Those generally were gastrointestinal or minor lethargy that usually resolved within 24 hours. Many of the vets also reported that these safety observations were confounded by the fact that they had to capture and stress the animals in order to vaccinate them, which also generates mild adverse events. Now, once the issues in mink farms became highly reported, the USDA issued a notice saying that they would accept a regulatory pathway to licensure for use in mink. That was when we pivoted over and did the studies in mink to seek a conditional license, which was issued in 2022. And we then had a donation program where we made available about 80,000 doses of the conditionally licensed vaccine to zoos, aquariums, conservatories, and some other animal organizations. That donation program, we've now stopped receiving requests, but we're still teetering the last few distributions out on that. And in this case, we have just under 110 organizations, zoos and aquariums in the US, a few conservatories, about 27 international zoos, conservatories, and some other organizations. And you can see the distribution of where those organizations are around the world. The title moved a little bit there, but one very exciting thing that we're able to report is this is the first case that at least we're aware of in the history of vaccinology where a single vaccine formulation has been used in over 300 different animal species. A few of them are shown up here. From the primates, felids, canids, many of the mustelids. These 4 groups make up the majority of the animals vaccinated. But then some other more minor families and species, including even dolphins, which was interesting. I was very interested to find out that even like manatees have been reported as being positive. Now, a few of the the taxon advisory groups have done their own studies separate from us. And this paper has been submitted for publication looking at serology in goats. This is the Caprine Taxon Advisory Group. And they were able to, at various time points, getting serum samples from these animals, were able to show a nice serum neutralizing titer. And their last time point was 180 days. And they were still seeing some serum neutralizing titers, although some of these larger goats, they start to wane off about 180 days, suggesting you might need a booster in those larger animals at about 6 months. Of course, remember, this was a vaccine that was developed for ferrets and mink, so much smaller animals compared to a very large goat. And then this is an interesting one from a single zoo, which I can't identify what the zoo was, but they had a number of different species, and they were able to get serum samples at opportunistic time points. We aren't able to show a line graph, but you can see when those opportunistic time points are scattered around, and when they were able to get the samples, they were able to look at serum neutralizing titers. And this one animal, this is a cat, a big cat, out at about 220 days, was still had a very nice high serum neutralizing titer. And I think that was my last slide, so I'd be able to take any questions.
Thank you, John. I'll start it off with the first question. My question would be, do you have information on how good the vaccine protects against virus excretion in one of those species?
Well, unfortunately, we didn't do the challenge work at Colorado State like our competitors were able to do. So we don't have a study showing the vaccine impact on viral secretions. So I don't have that data. What we do have is a lot of anecdotal data from the zoos regarding vaccinated animals. There was a few big cats that did get infected. In that case, the symptoms were very minor and lasted usually about 3 days.
And you're not planning to do such studies on that sort?
Not unless the regulatory authorities pivot in their positioning on licensure for companion animals.
Okay, thank you. Any other questions? Yeah.
Yeah, I'm curious, John, with such a range of the facilities that got vaccine and the range of animals that got vaccine, what did they tell you ahead of time? What were their motivations? I mean, full disclosure, on a project where we've gone to some of these zoos and asked when they had it, but it's a small subset. And the motivations seem kind of literally and figuratively all over the map. So what did folks, what were they asking for?
There was a wide range of discussions that we had with the zoo vets. Remember during the pandemic, there was very little information out at first. Then we started to have comparisons of the ACE2 receptors that were indicative that the host range may be pretty broad. Then dogs and cats, reports came out, and then you started trickling all these other species. So we also knew that the different variants may behave differently in different species. So nice work showing susceptibility. Remember, the zoos were literally shut down during the pandemic. They were very worried about their animals, and you started to see some fatalities reported. So for them, they were having to maintain social distancing from their animals, so they couldn't care for them like they would like to. And they were mostly focused on trying to look out for the health and well-being of their animals in a very fast-moving, unknown environment. Each organization did their own risk assessment. So what are their practices in terms of their interaction with the animals? What's the vaccine status of their staff and of the surrounding community? What's the transmission? And how many animals do they have, and what's the health status of them? Because just like humans, animals with comorbidities are more susceptible than very healthy young animals. So they were looking for opportunities to be able to reduce the risk of their animals getting infected.
Thank you, John. This is a great talk. I have 2 questions. One, can you talk about the expression system that you use for your recombinant protein?
Yes. So we used a proprietary eukaryotic-based system. We use it for a number of other monoclonal antibodies and also for other vaccine antigens. The protein was then purified by column chromatography. So it's a very highly pure recombinant protein. I showed you the electron micrographs. We were able to verify that it's in the correct pre-fusion conformation. And we can express it at very high levels. We were able to get up to about 2 grams per liter yield.
And then my second question is, wow, I mean, like you said, I don't think there's ever been, a case where one vaccine has been used across so many species, but I think you mentioned that it was, you used the mink dose, right?
We used the
mink dose, so it was a dose that was formulated for mink.
Yep. Well, originally it was planned for dogs and cats, but
okay.
it was the same dose as what was used for mink.
And you mentioned that you were looking in zoos and much larger animals. So how was that addressed? Was this, were the zoo veterinarians thinking, under a veterinarian-client type relationship that they could use a different dose, additional doses?
So
Yeah.
Good question. Very good question. So at the 300 different species, very large variation between some very, very small animals and your silverback gorillas. So we recommended to use the same dose size, but zoo veterinarians are very resourceful. They can also use off-label based on a reasonable scientific knowledge. We did have reports back that some vets on very, very small animals used a half a dose. For very, very large animals, we had some vets tell us that they used a double dose. We had some vets say that they used, instead of a 2-dose series, they used a 3-dose series. So some vets were a little more resourceful. Many of the larger or more susceptible animals, the zoo vets came back and said they wanted to redose at like 6 months, just to be sure. Some cases without any serological data. Some of the taxon advisory groups that I showed there were actually looking to develop data to guide their vets with.
It's a very new world for me, this one, but could you imagine a situation where a veterinary vaccine company would make a vaccine and it would go into humans?
Where a veterinary vaccine would go into humans?
Yeah, so you're a...
So very, very different regulatory authorities.
Yeah, but a pandemic and...
we use basically the same outline of production that's used in the human recombinant protein that's now coming out. It's literally the same, but the regulatory and oversight is very, very different. We have no plans whatsoever to produce vaccines for humans. You'd probably have to talk to the USDA and FDA and see what their plans are, but we don't have any plans for that.
There are human vaccines, but imagine next pandemic, and it would be good if the vaccine manufacturing communities were joined up, I guess, to maximize our chances of making vaccines as quickly as possible.
Yeah. Now, on the diagnostic side, many of the veterinary diagnostic labs turned over and became CLIA certified and did diagnostics, but from a vaccine production, we don't have any plans to do that. Cyril?
I was just going to add on that. This is actually a good question. During the pandemic in the U.S., in the federal government, they were looking at capacity for production. So they were actually looking at veterinary manufacturers that could produce the millions of doses that would be needed. So, I mean, it's never really been done, but I think there's more and more thinking about, in the context of a pandemic, what could be done more quickly by working together. So hopefully we won't have to face that again, but you never know. I think that was a good question.
Yeah, COVID-25.
Not just on the production side, but the development. You have a different vector system, a different way of producing, and maybe your vaccine happens to be better than someone else's.
Yeah, we were actually in talks with the company that's producing the recombinant human vaccine now, so they kind of mirrored our process as well. So it's very similar.
I'm Zoltan Penzer from SIVA, also a vaccine producing company. So I think it's a very good question, but I think my colleague already answered it, that it's completely different regulatory background. I mean, the factories are different. And the other thing I would say that, normally the capacities are quite well utilized in the industry. So from the moment, let's say, we would start to produce a human vaccine, which will never happen, I'm sure. There would be lots of clients or customers or farms. Because we cannot stop them because the supply is normally quite tight all over the world. So then we won't have anything to eat. We need chickens, pigs, and protect it from. Yeah. So it's a bit tough question. Yeah.
A discussion for tomorrow afternoon, perhaps. Yeah. I think there's one quick question here from Dallin.
Is there another question? Yeah. Oh, Dallin. Okay, that's the final question then, okay?
I'll sneak in two. One is quite simple. So I don't know anything about vaccination in zoos, but is it routine? Are they vaccinated against anything else, or is this completely novel?
The zoo vets have regular ongoing vaccinations against diseases that are appropriate for that species. Obviously, an elephant is going to be very different from a lion, but they do regularly vaccinate. Most all the time, those are off-label uses because I think for both SIVA and Zoetis, we can both say there's not a massive market for say a dolphin vaccine, or even a lion vaccine and others of such. So they usually use off-label.
Okay, the more complex question would be, I think a lot of the animals in zoos are actually born in captivity nowadays rather than captured. I think that probably varies a little bit on the type of animal, but I wonder whether or not the information that you're gathering in captive animals where the serological situation might be quite simple in comparison to wild animals, whether or not that affects the sort of potential interpretation of your data for vaccinating wild animals. So I'm thinking, perhaps with SARS-CoV-2, the susceptibility of like large primates was probably not huge, but I wonder if there was a virus that could kill gorillas, for example, we might want to vaccinate gorillas, and whether or not you could translate your data.
We did have a number of conservatories request doses. So those aren't wild animals, but they are sort of captive wild animals because they're not held in very in pens in a zoo situation. They are in a very large open landscape. So a little bit different, but the other thing is this is intramuscular or subcutaneously delivered, which would be a very difficult situation for a large-scale wildlife vaccine where you'd probably want more like a bait vaccine that would be orally delivered.
Thank you.
Okay. Thank you, John, for a very interesting presentation. Well, herewith I would like to close the session of presentations. Thanks to all the speakers. And I'm looking at Sharon for some final announcement, maybe.
Just a couple of quick announcements. So we've put up the slides from some of our sponsors here today, and I think we've already mentioned them. Louise has asked people to take a look at the Royal Society of Biology table. We've got the Microbiology Society that has funded the prizes for the early career researchers, which we'll be announcing tomorrow afternoon before the workshops. We've mentioned about the planning for the workshops, which we're going to do shortly, and a plug for the upcoming UK ICN meeting, which is the annual meeting which will be held in Slovenia shortly, and we'll also have a big data session the day before, so it's a 3-day meeting. That's all on the UK ICN website, where you can become members. As I said, free of charge, and then you'll get all of the mail shots, our seminar series, which runs on a monthly basis, and all of the other benefits of part of being the international coronavirus group. I'm going to ask our chief sponsor to come up and say a couple of words for us this afternoon. So that's Martin Mitchell from SIVA. He's interviewed quite a number of you today, and I know that some of the recordings are also going to be up on YouTube on our UK ICN site, and we will make the program and abstracts available as a PDF via that portal. And then after Martin's given us a couple of words, Louise is going to tell you about our drinks reception and the dinner for this evening. You have got a little bit of a break in between, but hopefully you can use that to finish looking at the posters and networking amongst yourselves. Martin, can I ask you to come up? Thank you.
Took me by surprise, I wasn't. I thought I was going to talk this evening at the dinner, so...
Just a short one.
Yeah, well, on behalf of SIVA, I'm delighted to be here, and we have Zoltan Penjes, who will talk tomorrow, who's our head of, not R&D now, I&D for biologicals, and I know that he's known to many of you. You know, my colleague John here from Zoetis, I've been interviewing many of you, and a lot of the questions were really directed towards pandemic preparedness. What have we learned? I know that we committed to become a One Health company about 12 years ago, and we're really examining the way that we do things to really understand whether we work really in a One Health way. And even within our company, which is we've got just over 6,000 employees, it's not joined up. Nobody understands what's happening. There's lots of things that are happening, and we are working in a One Health way, but we're not doing it systematically. And I think from the guys that I've interviewed so far, you all see real value in a network like this, and especially where we've all been confined to TV monitors or whatever else. And yes, we interact, but what it lacks, and probably what artificial intelligence will lack in the future, is that humanity. And what events like this do is bring people together, and the discussions that you have over coffee or dinner are just, well, probably more valuable than a lot of the discussions that we could have otherwise. So it's been really great to be part of the planning team with you, Sharon, Louise, and I can't mention everybody, but we're really just delighted to be here and to help you facilitate this meeting. So please enjoy the rest of it. Thank you.
Thanks. So just a little bit then about the this evening. Obviously, you've a little bit of a break now, as Sharon said, so you can have meetings, look at the poster, go a nice little walk in the grounds if you want, or go to a hotel if you need to, if it's near enough by. But for this evening, you've probably noticed they've set the tables out really nicely with candles, etc. So the dinner will be where you had your lunch, except it will be a sit-down dinner and food will be brought to your table. So just to ensure that those with you have dietary requirements, the Queen staff will ask you when they come to the table if there's somebody at the table has requested a particular meal outside the normal one that's on the menu. So please let them know, and they're usually extremely good at this. As I say, there will be a drinks reception at 7 o'clock before that, and then the dinner will start at 19:30. So I hope that everyone enjoys that and has a little break now. Thanks very much.
I hope you enjoy this evening, and we'll start again tomorrow at 9 o'clock for the wildlife sessions. Valeria, would you like to say something now? Oh, just reminding all of those involved in workshop coordination just to stay here for a quick 10 minutes for planning for tomorrow. Thank you.